So this is a screencast that's mainly about blood. And so I want to talk to you about the different kinds of blood cells, paying particular attention to how they're involved in immunity, so that you'll be able to relate this information to both innate and adaptive immunity. So when we think about blood, it's technically a connective tissue. And what that means is that it's a set of cells that are embedded in some sort of extracellular matrix. Your bones are connective tissue, and you have these little cells called osteocytes, and they're surrounded by an extracellular matrix of calcium carbonate. In the case of your blood, you have red blood cells and white blood cells, and those cells are embedded in an extracellular matrix that is the plasma. And so these cells are contained within the plasma, and these blood cells are created from bone marrow-based stem cells that are the same cells that produce your red blood cells, are also the stem cells that produce your white blood cells. So if you think about stem cells are these cells that can become a wide variety of different kinds of cells. So there's a blood stem cell in your bone marrow, and this blood stem cell can differentiate into either myeloid stem cells or lymphoid stem cells. The myeloid stem cells are the stem cells that give rise to your red blood cells, but they also give rise to things like platelets and macrophages and granulocytes, the eosinophils, basophils, and neutrophils. These are the kinds of cells that are largely involved in the general inflammatory response. They migrate to areas where you have an infection. They release chemicals that are designed to degrade the organisms that are causing that infection. They also secrete chemicals that cause an inflammation response, that cause your tissues to swell up and localize that infection to a particular area so that it doesn't spread throughout your body. And then it also recruits things like macrophages to come in and absorb those pathogens and destroy them. The lymphoid stem cells, on the other hand, produce a lymphoblast, which can differentiate into B lymphocytes, T lymphocytes, and natural killer cells. And these are the things that are primarily involved in what we refer to as the adaptive immunity, this pathogen-specific immunity that we talked about in the last screencast. So if you think about animal cells in general, the things that I want you to pay attention to are the membrane-bound parts of an animal cell. So eukaryotic cells with a nucleus, are made up of a plasma membrane going around the outside of the cell. Then you have cytoplasm, which is all of the stuff on the inside of the cell. And then the cytoplasm is made up of dissolved substances, sugars, proteins, lipids, etc., etc. And then also a set of organelles. Some of these organelles are membrane-bound organelles, like the nucleus, the endoplasmic reticulum, the Golgi apparatus, mitochondria, lysosomes, endosomes, peroxisomes. And then we have non-membrane-bound organelles, things like ribosomes. And ribosomes are involved in protein synthesis. They're the dots that are on the outside of this rough endoplasmic reticulum, right outside the nucleus here. And ribosomes are involved in protein synthesis, which you should remember from the information about sickle cell. When we think about the plasma membrane, you'll see that it's made up of a phospholipid bilayer, as well as a bunch of proteins that are embedded in the membrane. And these proteins can sometimes have complex sugars and things like this attached to the outside of them. And so these proteins have a variety of different kinds of functions. And depending on the structure of the protein, that will dictate what its function is. In some cases, we have a transporter protein. This is basically like a pore in the plasma membrane, the phospholipid bilayer, that allows substances to enter the cell or exit the cell. We also have membrane proteins that are enzymatic in terms of their function and that they facilitate chemical reactions. Then we have cell surface receptors, which are proteins that receive stimuli on the outside of the cell and transmit messages to the inside of the cells. And so these are cell surface receptors. And these are going to become very important in terms of the way in which COVID, for example, infects our cells. Then we have cell surface identity markers. These are involved in self-recognition. In this case, this is a glycoprotein where you have sugar chains attached to the protein, and it's those sugar chains that act in self-recognition. Then we have proteins that are involved in cell-to-cell adhesion, making sure that cells that are adjacent to one another stay connected to one another. And then there are anchoring proteins that essentially anchor the internal skeletal structure of the cell, which is referred to as the cytoskeleton, and anchor those to the cell membrane so that those cells maintain their shape. This is especially important in things like muscles that contract. So when we think about moving materials across cell membranes, we can oftentimes do this with vesicles. And in this case, we have a bacterial cell where the eukaryotic cell will basically form a vesicle around that bacterium, bring that bacterium into the cell, and once it's there, more than likely destroy it. This is referred to as phagocytosis, and this is what macrophages do. So when we talked about macrophages earlier as one of the types of blood cells, one of the things that macrophages do is they engulf substances through the process of phagocytosis. Pinocytosis is the process of essentially forming vesicles around smaller objects like molecules rather than organisms. And you can bring different chemical types into the cell by essentially engulfing them in much smaller packets rather than larger packets that would contain something like a bacterium. And that's pinocytosis, the formation of these tiny vesicles. And then we have receptor-mediated endocytosis. This is a situation where a molecule binds with a receptor protein, and when they bind with the receptor protein, there are these molecules just under the cell membrane called clathrans, and these clathrans will then form a vesicle that contains the compound that is bound to the receptor molecules. And as it turns out, COVID takes advantage of this particular type of receptor-mediated endocytosis in order to get inside of the cells when it goes to infect them. So when we think about cell signal reception, proteins have receptors that act in different ways. In some cases, there are chemicals that open chemically gated ion channels. There are sometimes what we refer to ligand-mediated signal, where there's a particular ligand that once again sends a signal to the inside of the cell. And then there are what they call G protein coupled receptors, where something happens with one receptor and one part of the cell membrane that then starts a series of chemical reactions that then affects another protein somewhere else in the plasma membrane. So the ligand interacts with this protein starts a cascade of chemical reactions that then tells this protein what to do. And in this case, it's an ion channel. And then we have what are called toll-like receptors, which once again, it's a ligand that interacts with one protein. That one protein sets in place a very specific set of chemical reactions that then tells another protein what to do. So when we think about different types of blood cells will interact with other cells in different ways. And so with red blood cells, they don't really have any role in immunity. Their main function is to carry carbon dioxide away from cells that have generated it and bring oxygen to those cells. They lack a nucleus in mammals, and that's what makes them biconcave. That's why they look like a donut, is because they're thinner in the middle and they're thicker at the outside. And other groups, though, possess blood cells that have a nucleus. And so if you look, for example, at a bird or reptile red blood cells, you'll see that they are not biconcave. They actually have a nucleus still inside the cell. So red blood cells really aren't involved in immunity per se, but all of the rest of these things are. So when you think about granulocytes, these are called granulocytes because they have little granules around the outside of the cell and also inside the cell. And it's because these things are the cytokines and other chemicals that are going to be used to mount this general inflammatory response that we talked about earlier. So granulocytes include basophils and neutrophils and eosinophils. They secrete these cytotoxic chemicals that break down other cells, and they're also responsible for the inflammatory response. This part of the inflammatory response is also the allergic response. So if you go into anaphylactic shock, for example, it's these kinds of white blood cells that are leading to that particular response. And an anaphylactic reaction is just an inflammatory response that is on steroids, that is gone further than it needs to go, and actually is now risking your health rather than helping you respond to a particular infection. These cells also sometimes present antigens to other cells that are involved in the adaptive immunity process. This is the case with macrophages. So when we think of macrophages are once again these cells that undergo phagocytosis of things. They can phagocytize pathogens. They can phagocytize cells that have been infected with something like a virus. They can get rid of cancerous cells, and they're also involved in dismantling cells that are dead or dying. Then when we get to lymphocytes, these are the white blood cells that are involved in adaptive immunity. So they come in 3 different forms: lymphocytes, T lymphocytes, B lymphocytes, and natural killer cells. These things recognize and destroy pathogens such as viruses and bacteria. And these B lymphocytes primarily make antibodies, but T lymphocytes come in 2 different forms. One form produces antibodies. The other form is basically a cytotoxic form that is capable of causing cells to break down. And they kill pathogenic or infected cells and cancerous cells. And these lymphocytes can also signal other immune cells by presenting antigens to other lymphocytes like B lymphocytes. And you'll understand later what that means, but just put that in the back of your head for now. And the natural killer cells destroy cells that are infected with viruses, and they do this by prompting a cell to undergo apoptosis. We're going to skip this slide because I actually have a better slide later on when we talk about getting infected by a virus. And I think that's the end of this screencast, so we will shut that down. The next screencast will actually talk about how the virus, COVID virus, gets into you and accomplishes its infection.